my name is Erol Kavvas, and today I'll be talking to you about my paper titled A Biochemically Interpretable Machine Learning Classifier for Microbial GWAS. Well, the primary motivation for the study comes from these studies known as genome-wide association studies, GWAS for short. The basic idea in these studies is that people take thousands of genome sequences with corresponding phenotypic data, such as the color of our eyes or whether a certain microbe is resistant or susceptible to an antibiotic, and then apply statistics to these data sets to identify genetic associations, specifically the genetic factors that best explain these phenotypes. So for the example of Mycobacterium tuberculosis, the dangerous pathogen that kills millions of people, along with its drug resistance phenotypes, people have been taking those data sets and applying, statistical tests to identify the genetic factors that underlie drug resistance. However, these genetic associations are limited in their explanations. All we could say is that this genetic feature is associated with this phenotypic trait. It remains unclear exactly why or how these genetic variants actually alter the metabolic state or regulatory network in such a way that actually creates this resistant or susceptible phenotype. For the case of Mycobacterium tuberculosis, there is increasing evidence that there's actually a metabolic basis for the relationship between genetic variation and these drug resistance phenotypes. In fact, over 75% of these genetic variants are metabolic in nature, and there have been studies showing that many of these variants actually have metabolic selection pressures that underlie the resistance phenotypes. And so what we wanted to do in this study was develop a method that not only takes in these genomics and phenomics data types and gives you the causal genetic variants, but also provides mechanistic associations, in particular metabolic associations. Well, since our study was a methods paper, the following outline of our results were as follows. First, we described the method titled Metabolic Allele Classifier. We described this method. Then second, we validated our method using a held out data sets corresponding to 3 antibiotics. And then finally, we applied our method to 3 antibiotics to identify and recover the genetic associations, as well as provide new insights regarding their metabolic mechanisms. Since there's lots of results in our study, I'm just going to go over one case for the antibiotic, pyrazinamide. If you want to look more into these other cases, then go read the paper. So first, the method. Our method is named Metabolic Allele Classifier, MAC for short. Our MAC has 3 inputs. First, thousands of genome sequences of tuberculosis provided by the Patrick database. Second, corresponding drug resistance phenotypes also provided by the Patrick database. And then third and finally, the genome scale metabolic model of Mycobacterium tuberculosis that I constructed hella days back in 2015. So now for the method itself. We did what no one's done before. We used flux balance analysis as a machine learning classifier.
Yeah, believe it, dude.
So the metabolic allele classifier can actually be visualized as follows, similar to flux balance analysis as a polytope, where you could see to the figure to my left that the outer black polytope is the classical flux balance constraint, and then the inner blue and red polytopes represent allele-specific constraints that constrain the the resistant and susceptible Mycobacterium tuberculosis strain. So this is for a specific drug to the figure to the left. And so we have certain parameters here, one of which are these allele-specific constraints, as well as the coefficients of this hyperplane. And so we employ a variety of computational approaches to determine these that are derived from machine learning. For the second part of the results, we have the validation of the metabolic allele classifiers. Doing this was simple and straightforward and very common to testing machine learning classifiers. And so what we did was we trained these metabolic allele classifiers on a training set and then held out a test set of a certain amount of strains to see how well our machine learning classifier, the metabolic allele classifier, could perform on this held out test set. And so you can see from the figure to the left, we performed this for a variety of different training sets for different antibiotics, as well as a very large test set ranging to about 1,200 for a couple of antibiotics, all the way down to about 200 for pyrazinamide. What we found was that our method actually can get area under curve values similar to our previous machine learning approach, which means that our approach performs on par with current machine learning classifiers. Furthermore, we looked at whether we could recover the primary genetic determinants that are known for these antibiotics. And so we plotted the coefficients of this hyperplane I showed previously, and we found that the most heavily weighted coefficients actually correspond to the known primary genetic determinants. So in this figure, you can see that we uncovered the metabolic reactions that correspond to PNCA for pyrazinamide, which is the known primary genetic determinant, Thi for para-aminosalicylic acid, which is the known primary genetic determinant, as well as Cat-G for isoniazid. And so this is strong evidence that our metabolic allele classifier is actually recovering known biological features as well as accurately classifying strains as resistant or susceptible. So I've described the method, the metabolic allele classifier, how we validated the metabolic allele classifier, and now I'll briefly describe application of the metabolic allele classifier to the antibiotic pyrazinamide. Since these results are rather deep, I'm just going to focus on pyrazinamide, but you can look at the paper to get further information about the application to para-aminosalicylic acid as well as isoniazid. So the results are really about understanding the flux state. So when we apply the metabolic allele classifier, not only do we get these allele genetic associations, but we also get these metabolic associations. And these metabolic associations come in the form of associated metabolic fluxes, which really are these pathway activities, which come from these steady state constraints. And so for pyrazinamide, the first thing we did was look at, all right, what flux states differentiate resistant and susceptible tuberculosis strains? And we took those flux states and performed pathway enrichments. We found that the most enriched pathways corresponded to thiocerol biosynthesis, as well as nicotinamide and nicotinamide metabolism. So this is actually consistent with the literature where nicotinamide metabolism is the most affected pathway, which really comes from deleterious mutations in the gene PNCA. Now, the thiocerol biosynthetic pathway is less obvious, but there's been research showing that that's also highly enriched in pyrazinamide. So now we decide to go in a little deeper and see, all right, why is that? And so the next result are really these allele-specific hypotheses. And so currently in the literature, people just say, all right, these alleles are associated with resistance. Now, as you could see, we're not only identifying the alleles, but we're actually computing their metabolic effects, and so the functionality. So in the case of PNCA, which encodes the nicotinamase reaction, we see that the susceptible dominant allele, the one colored blue, has a higher relative flux than the two resistant dominant alleles. This means that the resistant dominant alleles are deleterious, or mean that that now the reaction encoded by PNCA is non-functional and can't run. This is consistent with the literature that shows that these mutations are actually deletions, which make a non-functional enzyme. New to our study is this association with the gene ANSP2, which encodes a asparagine transporter. We find that the resistant dominant allele actually increases flux of asparagine. So that's a new hypothesis from this study. Finally, we identify PPSA alleles, which encode reactions within this thiocerol biosynthetic pathway. We find that the resistant dominant alleles are actually increasing flux through this pathway. So now we could take a full pathway view of this perspective. So in the paper, we also provide these large pathway maps, and you could see what's going on here. And so we actually traced the effects of these ANSP2 alleles and found that they're actually increasing the coenzyme A balance, which in turn is probably affecting the thiocerol biosynthetic pathway. So there's previous literature showing that coenzyme A is actually implicated in pyrazinamide resistance for different reasons. But in this paper, we implicated it through ANSP2, which is a novel hypothesis and warrants future experimental investigation. Well, genome-wide association studies are still one of the most dominant forms of investigation for understanding the genetic basis of phenotypes. From understanding breast cancer to pretty much any sort of phenotypic trait we have, GWAS is still the most dominant approach. And so not only does our method do similarly as GWAS, but it actually extends the hypothesis reached by GWAS. And so our approach is important because now, instead of just providing genetic associations, we provide metabolic associations that can help provide deeper insights into actual causal mechanism underlying phenotypes. This has wide implications from medicine to agriculture to various fields, where now we could understand and better design drug treatments or design better synthetic organisms, etc. In the case of Mycobacterium tuberculosis, our results identified or recovered genetic associations previously shown, but now provided actual metabolic hypotheses for why these exist. So people in the TB research community can now take these results and say, okay, maybe we should treat pyrazinamide with these kinds of antibiotics so that we can better combat this organism. Or maybe provide these sets of nutrients to a person who has TB so that they could better heal and deal with this disease. We expect our approach to be widely used in the field of GWAS and to further provide new insights into any type of genetic associations where a genome scale model of the organism is available.